much the organisers of this conference, especially for having to rearrange things last minute. It must have been a massive undertaking and a massive nightmare. So I'm really appreciative of that. And also really want to thank Liz for allowing me to present this to you and also involving me in this research and being so supportive of my own research on native bees. So big shout out to Liz. So today I'm going to be talking to you about using a novel method to record insect visitation to flowers and our findings on this. So insect flower visitors are key parts of biodiversity and of ecosystems. By recording insect visitation, we can understand floral preferences, pollination ecology, competitive interactions, how these interactions change with environmental factors like temperature and phenological patterns, climate, and also how they might change with management, and understanding the different types of visitors that influence visit flowers can enhance our understanding of the evolution of plant pollination networks and provide us with a greater understanding of how we can better manage ecosystems. And that there was a picture of an amygdala, which is most people's favorite native bee. It's a buzz pollinator, and so it visits flowers with poricidal anthers that require buzz pollination, which is something that honeybees actually can't do. Now, the opportunities of using video recordings over traditional insect monitoring methods are multiple. So the great thing about using video recordings to record insect visitation to flowers is that it can be standardized. So as part of my PhD research, I looked at a number of methods, including observational, sweep netting, bee bowls, and vane traps. And I found firstly that the passive methods were vastly inappropriate, but sorry, not COVID. But for sweep netting, which was the best way to catch bees, it requires actually a lot of skill. And so for some people, they're going to be quite terrible at catching bees. And so with video recordings, we can standardize this insect monitoring method, and it allows cross-verification because it's recording, different people can look at it and we can make sure that we've got the same results. You can also record at different canopy heights, whereas with observations and sweep netting, you're limited to basically a meter or two. You can also record at multiple locations simultaneously, which is something I couldn't do because I can't clone myself. And you can also quantify flower area because you've got this recording. You can look at the area of the flowers and various other metrics that aren't possible or take a lot more time if you're doing it in real time. And you can accurately measure foraging duration and flight patterns and using sophisticated video recording technologies, which is something that we are investigating. You can look at streamlining this process and getting some really sophisticated analyses. But there are also some limitations, and we have to be transparent about these limitations. So using video recordings, it's impossible to identify the majority of visitors to species level. So as you can see here, this is a collection box of my native bees, and the differences between species are microscopic. They can be the shape of the wing veins, they can be the genitalia, which is clearly something you can't pick up on video recordings. There's a limited field of view. It depends on the angle of the camera. It can take a very long time to process all the videos, but we are looking at a method to overcome this, and there's a limited battery life. So our experimental design is that we undertook this research on North Stradbroke Island, which is a tropical paradise, and it's a really beautiful habitat. Recordings were made from 72 plants from 24 plant species over 6 field trips over 12 months. And 4 plants from 1 to 2 plant species were recorded each day over 3 days per visit. And this involves setting up a pair of GoPro cameras, mounting on a stereo configuration at each plant, and recording for 60 minutes, because as I mentioned, there's a limited battery life, starting 1.5 hours after sunrise, then again at midday, and 2 hours before sunset. Now, for the analyses, I looked at bipartite networks, which looks at patterns of interactions between plants and their visitors. And this can reveal a lot about the structure and function of ecological networks. You can look at how robust they are to extinction, look at how connected, how many interactions they are, and the shared interactions between species. In particular, there's this index called the potential for apparent competition, which is basically a really neat way of looking at resource overlap and therefore the potential for competition. And with our analyses, we looked at both the number of arrivals, which is your typical analyses for visitation networks, as well as another metric called max N, which has not been used before in plant pollination studies, but is used a lot with this sort of video recording software for underwater analyses, which is where this design was inspired for. Now, we recorded 1,565 individual flower visitors, comprising 93 distinct taxa. So there's a very high diversity of taxa that we recorded visiting these flowers. In terms of number of arrivals, 4,604 visits by volant taxa observed. And the European honeybee dominated. Australian native bees and Diptera were the next most frequent, and this is very apparent from the bipartite network. So you can see lots of visitations, honeybees visiting a lot of the plants, but a really complex and healthy ecological network on this island. Now, max N, that's a bit different because it's basically allowing us to look at the maximum number. It can underestimate, whereas the number of arrivals can overestimate. But with max N, we were able to record ants because they don't basically arrive. Max N has allowed us to record that. And ants actually comprise now a major component of the visitors to flowers. Now, something to be aware of is that just because something visits a flower definitely doesn't mean it's a pollinator. So ants could have been nectar thieving or pollen thieving. So potential to disrupt, I guess, the actual pollination networks by deterring actual pollinators. Now, I also looked at variation among the plants in their attractiveness, and one of the plants received almost a quarter of visits, so it was very attractive. Banksia was also very attractive, which is sort of a bit, I guess, a bit unusual when we think of Banksia more of a bird-pollinated plant based on its morphology, but it was very, very attractive. And then the Metaceae were also very attractive as well. There was, as you can see from the networks, which are the attractiveness of a plant is indicated by the width of the bar. There was a lot of variation in how attractive these plants were for the different groups. Now, visitation duration, there were significant differences. Beetles, they visited a lot longer than the other taxa, in particular, the unknown taxa, so those are the ones we couldn't identify from the videos, and the Diptera, the flies, and that sort of is in accordance with basically our observations. Beetles tend to be quite slow, spend a long time with flowers, whereas flies zip around. And also there was variation on how long taxa spent on different plant species. So particular species of Melaleuca, the visitors tended to spend a lot longer on that species, and it could be that based on the flower that you can see there, it's got a lot of pollen and a lot of nectar. Now, what I did was I wanted to know what was the key determinants of visitation. So I used a multivariate model selection procedure, and I put in all the potential explanatory variables of what influences visitation. I found time of day was a key component. So there were a few visits in the morning. This is mainly driven by honeybees, and this makes sense. You know, they like being ectotherms, they need to be warmer, and so they were foraging more in the afternoon and in midday periods. But the most important factors influencing number of visits. So despite the variation in plant species, this wasn't the overarching factor. More flowers meant more visits, which sort of makes sense, more resources for all the insects. Temperature, again, making sense given that these are ectotherms, so their body temperature is reliant on that of the external environment, so more activity when it's warmer, and activity decrease with wind speed. Again, it's harder to forage when it's really windy. So environmental factors emerge as being really important in influencing insect visitation to the flowers. Now, network properties, I found that generalization, they were not very specialized, these networks. So most of the insects visited a variety of flowers, and likewise the flowers were visited by a variety of insects, and they were also really nested, and nested is a property that confers network stability, and it's a sign of healthy pollination networks. And these were, so they were much less specialized and more nested than those that I calculated from urban bushland remnants and residential gardens. This could be due to the broader taxonomic range of insects in the present study, whereas I just focused on bees, and the lower taxonomic resolution here though. So these were really broad taxonomic categories, whereas I used genera, but it also could be a property of these networks compared with the urban networks. So remember, this is, almost pristine habitat here. Now, potential for apparent competition. Apis mellifera definitely dominated. So these colours show the more red it is, the more potential for apparent competition. So it's looking at the column species on rose species, and Apis mellifera, tended to have a high potential for apparent competition with all taxa, more so than any of the other taxa, in particular for the specid wasps and Hemiptera. But when we look at the actual effect of honeybees on insect visitation, there was a positive correlation, and this is because the definition of competition is when there's resource overlap, which there definitely is between honeybees and the other visitors, but also when resources are limiting. So in this, really pristine, healthy ecosystem, there were abundant flowers, and it appears that resources weren't limiting, and honeybees and native bees and the other insects were responding favorably to the same conditions. So there's a positive correlation there. Although it was, a fairly weak one, it was definitely significantly positive. This contrasts with my research on honeybees in urban areas where, although there's again much scatter, which is normal for ecological systems, there were negative correlations between overlap and the abundance of native bees. So this is again drawing attention to how important it is to have a lot of native vegetation. In urban areas, they're dominated by exotic plants, and so this is a real threat to native bees regarding competition with honeybees. But in this island ecosystem, because there's so much native vegetation, this is a really healthy ecosystem for coexistence. And one of my papers, I found that ecological traits really influence whether honeybees compete with native bees or not. But when I looked at the different taxa, the insect taxa, and their relationship to honeybee abundance, they all exhibited either no association or weak positive association. So again, no evidence for actual negative impacts of honeybees on any of the taxa, despite high overlap. So in conclusion, this was a novel systematic monitoring system, one of the first time it's been applied in a really important honey production area. So we've been hearing a lot about leptospermum today, and Stradbroke Island is a really important place for medibee, which produces honey from leptospermum, and it's also a really important conservation area on North Stradbroke Island. And as you can see, they've got some really amazing native bees. There are drawbacks to the system. It's limited by the lack of species and therefore trait level information. So as I mentioned, The categories were very broad, like native bees, whereas in my studies I've passed it down more to native bee genera. And this can, when you're aggregating species all together, sometimes this can mask some important ecological interactions, but it does allow, you know, standardization and including a whole range of taxa in looking at these visitation patterns, and some really interesting ecological information was gained through this study using this technique. And one of the key conclusions was how the high proportions of native vegetation means resources aren't limiting here, so there appears to be no competitive impacts between honeybees and the native insect fauna in this ecosystem. So thanks everyone, and open to questions.